Hello students, in this video we are going to study about types of asexual reproduction. So let us start. Very first we have the fission. There are various modes of asexual reproduction: fission, fragmentation, budding, regeneration, vegetative propagation and spore formation. So let us start with the fission. It occurs in a single celled organism. This process takes place in unicellular organisms basically that belongs to Protista and Monera. It is of two kinds, binary and multiple fission. Here we have binary fission. So as you are able to see that here we have a prokaryotic cell, either it is a bacteria or any other prokaryotic cell which undergoes binary fission, divides into two same sized organisms, same size daughter cells. The parent cell divides into same size daughter cell. In amoeba, the cell division or the splitting of cell takes place in any plane. From any plane, the amoeba divides. But leishmania, that causes a disease named as Kala Azar in case of human, divides longitudinally. Okay. Leishmania bears whip-like flagella at its one end, and the cell division takes place in relation to the flagella. Here you are able to see the diagram of binary fission in amoeba. First of all, we have the parent cell. This parent cell divides. In this case, we have first of all nuclear division. The nuclear division takes place. Then we have cytoplasmic division. Then the cytoplasm divides. First of all, the nucleus divides, then the cytoplasm divides, and resulting in the formation of two daughter cells. So here we have the diagram of binary fission in Leishmania and Paramecium. As you are able to see that Leishmania divides longitudinally and the Paramecium divides transverse vertically. Okay. So multiple fission. In multiple fission, a single cell divides into many multiple daughter cells simultaneously. Example, we have Plasmodium and amoeba. As you are able to see here, in case of amoeba, we have a single cell that contains a nucleus inside it. This nucleus first of all form many daughter nuclei which are surrounded by cell wall, cell membrane. And these nuclei, they form a large number of daughter cells. When it matures, it is surrounded by a cyst wall. When the wall ruptures, then these nuclei, they comes out from the body of a parent cell. Next we have regeneration. Regeneration is the process by which organisms are capable of generating or developing their lost part of the body. So it is defined as a process of formation of new individual from its lost part. Simple organisms utilize this method to develop the complete body, while the multicellular organisms or the complex organism which have the organ system level of body organization, they are able to develop the lost part of their body. For example, muscles, skins. You might have heard about the house lizard which sometimes lose their tail, and the tail regenerates again. Similar goes with the starfish also. So if the tail of a house lizard is cut, then the missing part develops again from the remaining part of the tail. Regeneration may be defined as a natural ability of the organism to replace the worn out parts and repair the or renew the damaged part of the body. Sometimes the whole body will be regenerated in case of lower organisms. Here we have the example of planaria. When the body of planaria is divided into two parts, then the two parts develops into the complete organism. Regeneration can also take place in hydra. Here you are able to see that this is a hydra, parent hydra, which when divides into two parts, the adult hydra, when divides into two parts, then the two fragments or the two segments divides into the complete organism. And here we have the house lizard. Now second, that is binary. Third one is binary budding. After binary fission, multiple fission, spore formation, we have a process named as budding. Budding means when a small projection or a small bud, or we can say it is a lateral outgrowth that develops on the body of the parent hydra. As you are able to see, this is the parent hydra. It develops a bud. This bud, when matures, then it detaches from parent body and it develops into a new hydra. So it is seen in fungi, multicellular organism. The parent body develops a lateral outgrowth. The nucleus divide and one of the daughter cell passes into the one of the nuclei passes into the daughter cell. The bird on maturation gets separated from the parent body and develops into a new individual. Thus, budding results in a formation of daughter cells of unequal size. It might take place in yeast, fungi, hydra, and multicellular animals. Next, we have a difference between regeneration and fragmentation. Regeneration and fragmentation, these both are the types of asexual mode. But in case of regeneration, we have the development of a part from the lost part, broken part, like lizard cast off their tail to escape the predators and they generate it again. But fragmentation is a mode in which there is unintentional cutting of the body. The body is cut down accidentally in regeneration, but in fragmentation, it is unintentional cutting of the body, which each part or each segment, or we can say each fragment, develops into a different organism. Next, we have spore formation. The next mode is spore formation. It can be seen in fungi. It is very common in fungi. In case of fungi, there is a thread-like structure that develops on the bread piece or bread mold, and the structure that is termed as hyphae. And this hyphae that bears a sporangium on its sporangiophore. This sporangium bears spores in itself. The bulge structure, the ball-like structure sporangium that contains the spores which develops into new fungi. You might have seen on the bread, there is a green color structure that is a rhizopus fungi. The spores are covered by thick walls which protect them when they come in contact with the moist surface and begins to grow. Next, we have vegetative propagation, the development of the new plant from the vegetative parts of the plant like roots, stem, and leaves. We have this under appropriate condition is termed as vegetative propagation. The plants can be raised. There are various advantages of this process. We can raise the plants that can bear fruits and flowers earlier than they produce than those produced from the seeds. So we can produce the plants without using seeds by this method. This method can be possible to produce the plants like banana, orange, rose, jasmine, which lose the ability to produce the seeds. The next we have another advantage that it is very cheap method and the plants produced are genetically similar to their parents. The very common example is when the in case of bryophyllum leaf, the buds produced at the notches or the leaf margin develops into a new plant when they fall on the soil or develop into a new plant. Here we have runners. Runners, it can be seen in case of mint. You might have seen that when you plant a mint in soil, the it spreads over a large area. Why? The reason behind that is it undergoes the vegetative mode of reproduction and the underground stem, resulting in the formation of large number of plants. So these birds grow into a new plant. Next, we have the vegetative propagation by roots. The swollen roots you might have seen, they are tubers that bears bud which develop into new plants. You might have seen in case of potato, okay. In case of sugar, there's a sweet potato. So these are the we can see the potato tubers which is in a formation of a new plant when dipped in the soil. Stem cutting. Sometimes we have two stems and they are termed as scion and stalk. When these two stems are joined together, but the condition is there that they belongs to a same family, they belongs to a same species. So cutting is a part in which the part of a plant is cut. Parent plant is cut and shoot with the leaves attached are usually used in this. So new roots and the leaves develop from the cut part and the shoot is cut at an angle. Now layering. What do we mean by layering? When we'll cover the when we'll bend the plant and put it in the soil again, the tip of the shoot remains above the ground and the roots eventually develops into a new plant. So these plants can then be separated. It can be seen in case of jasmine. Now here we have grafting. In case of grafting, we'll take two stems. One is known as scion, another is known as stalk, and these are joined together. Two plants are used to develop a new plant with a combined trade. Okay, like we will do in case of hybridization. So in grafting, one part is termed as scion, that is above part, above the ground, and this stalk scion that is attached to the stalk, which is rooted part of the another plant. So in this way we'll have a hybrid plant. Thank you.